Life is motion. Motion of individual organisms or the motion of the cells that constitute the organism. For example, when the developing organism grows, the cells divide to multiply. What is the source of this cellular motion? It's driven by the cytoskeleton, a network of interconnected dynamic filaments. Myriad minuscule proteins comprising the cytoskeleton self-organize into molecular machines which propel cellular motion, enabling, for example, cell division. How do the individual players, the individual proteins, work together to generate motion? In our lab, we use a combination of biology and physics approaches to answer this question. We use a bottom-up approach, assembling molecular machines from scratch. We isolate the individual proteins from cells and combine them to build functional units composed of only the essential proteins. Using this approach, we have full control over the system, which enables us to disclose the underpinning mechanisms that drive the movement in the living organism. We can thus visualize how cytoskeletal proteins form filaments, or how other proteins cut the filaments, how molecular motor proteins walk along the filaments, how they slide filaments relative to each other, and how they bundle filaments into higher-order structures. Using our cutting-edge equipment, we can manipulate single cytoskeletal filaments or proteins. We can visualize single molecules at work and measure the forces they exert. The goal of our research isn't just to visualize the processes that lead to cellular motion, but to understand the underlying physical principles. When we understand these principles, we will be able to predict and modulate the cellular motion. We thus hope to open new horizons for the design of novel diagnostic or therapeutic tools.